Hi, everyone. That one. All right. See who all is here. I was logged in twice, so I was getting an echo. My other computer doesn't have a camera, so I had to switch over to this one. I think, okay, we are recording. All right, and someone signed in. We'll give everybody a few minutes to see who else signs in. I think that's Jasmine. Let's see. All right, so Jasmine, I'll just wait a few more minutes for other people to connect. I'll just give them a few more minutes and then we'll start. And as we go through, this is being recorded. Right now you're muted, so if as I'm going along, if you have questions, just unmute yourself and jump in. Or you can post that in the chat box. What I'm going to be doing is just sharing my screen with you so that you can see we're going to go through just the course objectives for the chapters that are on this exam. And we'll go a little bit over the activity one, the questions on that. All right. Let's see. I know it took me a few minutes to get in here. Are you, do you have access to your email right now? I guess you do because, well, unless you signed in through D2L or got the invite through D2L. I don't have access to my email. And I don't know if it's a Trident Tech issue. I'm on campus right now, so maybe it's an on-campus issue. So let me know if you guys are getting this. All right, so if you have any questions as we go, just go ahead and, okay, oh good, so you have your email, good. All right, so I'm gonna share my screen with the information with just the chapter objectives. That really should be your main study guide when you're studying for your exams. Those bullet points on there, our list of learning outcomes. So we use when we build the exams. Even with my videos, even watching the videos, you do have to read the book. You know, that's just one of the things with college learning. You're going to have to read the textbook, even with all the other resources that are available. So watch the videos, watch those short videos by Dr. O. But do go through the book, especially take a look at, make sure you understand the images, the tables, those, the captions under them, describe them. There's a lot of detail that's covered in the book. A lot of times there are words that are on the objectives that aren't necessarily in bold in the textbook. So when you see something like that, you might have to look a little harder for those words. All right, so let's take a look at these. All right, so for chapter 1, and again, just unmute yourself and jump in if you have any questions, because right now while I have this sharing, I can't see the chat box. So if you type in a question, I won't see that right away. So just unmute yourself and jump right in. All right, so the first The first section, 1.1, this is pretty interesting, just kind of set the tone, gave you some background, and we come back to a lot of these same concepts. So how our ancestors improved food with microbes. They used microbes, bacteria and fungus, for a lot of different processes, from preserving foods to pickling foods, fermentation for wine and beer. So they used microbes in the process of preserving as well as enhancing the flavor. The causes of sickness, prior to the invention of the microscope, prior to being able to see these microscopic invisible things that could cause illness. Everybody got those. It was pretty interesting. The punishment from God for something bad air. But it was just the air around you happened to be bad, not necessarily that there were invisible microbes in it. And then you all did a great job in the discussion, in the first week's discussion, talking about the contributions of Van Leeuwenhoek with the first microscope that had the ability to see down at that microscopic level, at bacterial level, things that couldn't be seen. Even with other microscopes existed, but really they were just magnifying glasses. They weren't really at that level that Van Leeuwenhoek achieved. And then Louis Pasteur, who contributed all sorts of stuff, pasteurization being the one that we credit him with by actually naming it after him. But also he saw that microbes caused fermentation, and he contributed to vaccine development, like rabies vaccine. Tracy, if you have any questions, just unmute yourself and jump in. We're just going right through our learning objectives. If we just need clarification on, unmute yourself and ask. Stop me at any time if you need further clarification. All right. But stay muted until you have a question, just so we don't have a lot of background noises coming in. Thanks. Robert Koch, these are Koch's postulates. We talk about those more a little bit later, but he was the first one to associate or connect a specific microbe with a specific illness or disease. Taxonomy, I think we all went through classification pretty well. You don't have to really memorize a whole lot of those lower levels for different organisms. It's kind of what we used those when we looked at our parasites and our fungus. But the real levels you should make sure you know are the 3 domains. We have the archaea that we're not going to spend a whole lot of time on, but you should know how they differ from the other groups. So we have the archaea, we have the bacteria, which is where we spend most of our time when we talk about prokaryotes. But our archaea and our bacteria have a lot of similarities. They used to be in the same group until we got better technology, better techniques that allowed us to differentiate how they are different. So those 2 domains are both single-celled organisms. And then the third domain is the third domain that we have are our eukaryotes. So prokaryotes include archaea and bacteria. And then our eukaryotes are, this pop-up window keeps coming up. Wait for that blue circle of death so I can turn it off. That blocking. But our fourth group, the eukaryotes, those are cells like us, where we have a nucleus. So that includes the 4 kingdoms of plant, animal, fungus, and protist. So be very careful, because protist, prokaryote, we have a lot of these prefixes because the prefix means the same thing. The prefix adds to the base word. You really have to make sure you get the whole word, not just the prefix. So we have, where did that? So prokaryotes are our single-celled organisms that are simple, that don't have a nucleus. And our protists are a kingdom in the domain eukaryote. They do have a nucleus. So make sure we don't confuse those too much. Describe how, I'm sorry for that pop-up window in there. I can't get rid of it with that little blue thing in here. And I can't move it. I think this has something to do with our emails not working here on campus today. All right, so we're going to try to look around this. Scroll down a little bit here so they can scroll up so we can see what the next group are. Describe how microbes are named, how to properly write the scientific name. You all did that in our discussion forum. So remember, italicize. The whole underlining thing came because typewriters didn't have italics. Typewriters only had one font. That's the one you got. And so that's where that underlining process came for writing their names. Now we all have access to word processors, so you can italicize, even in our discussion forum. So make sure you use those tools to italicize in the discussion forum. All right, the types of microorganisms and acellular infectious agents. So our microorganisms are our bacteria, our prokaryotes, our protists, which are eukaryotes. Those are our single-celled parasites. Helminths, which are eukaryotes also, but these are worms, multicellular worms, and then fungus, and our acellular pathogens, viruses, and prions. Differences and similarities. So we've talked a little bit about archaea and bacteria, our two kingdoms. These used to be the same because these are single-celled organisms. They have similar structure, similar under the microscope. Yeah, I can't get rid of that. Sorry. Sorry. So they were classified together. Some of the distinct differences that have come about as we've gotten better molecular techniques is we can look at these and we see that their ribosomes are different. So archaea actually have ribosomes that are a little bit more similar to eukaryotes. Their cell wall is different. The cell wall of archaea is pseudo-peptidoglycan. It's like peptidoglycan, but not exactly. So it's fake peptidoglycan, pseudo. And the bacteria have peptidoglycan. So the archaea are really considered a transition, maybe closer to or midway between the bacteria and the eukaryotes, since they have a lot of similarities, like their ribosome structure to eukaryotes, but they are still simple organisms. So molecularly, they are distinct. One of the things with archaea is only recently identified that some of them are part of our microbiome, that they do live on and in the human body. None of them are known to cause disease. So we don't really spend a whole lot of time looking at those. Different types of cellular, viral microorganisms, infections. So that again was part of our discussion forum where we went through these, and we go in this in a lot more detail. So this was very general, just these broad bacteria, helminths, protists, fungus, and we go into that in more detail in chapter 5. And then provide an overview of different areas in the study of microbiology. So we have bacteriology, studying bacteria, mycology, studying fungus, protozoology, studying those protists, parasitology, studying our parasites, our frequent living helminths, and virology, studying viruses. Good so far? All right. That was a pretty good general overview. I like this chapter 1. It's interesting. It kind of sets the tone, and we keep coming back and building on that terminology. Chapter 3, we got specifically to the cell. So 3.1 was just a little more background, talking a little more about Louis Pasteur and how he disproved spontaneous generation. So spontaneous generation is also called abiogenesis. So Genesis, the creation, bio of life, A meaning not, from not living things. So spontaneous generation was that organisms just arose. And Pasteur demonstrated that all living things arise from other living things. So biogenesis. So it just sort of expanded a little bit of what we learned in chapter 1, looking at cells. 3.2 goes into how did we really figure out the basic unit of life is the cell and what cells are, and the cell theory. Some of the basic points of the cell theory is that all living things are composed of cells, either a single cell if you're a single-celled organism, or lots and lots of cells, and all those cells came from other cells. So through cellular replication, how we build populations, and that the cell is the simplest unit of life. And then if we look at the contributions, sort of expand our knowledge of what went into developing this. I liked in the discussion, someone brought up the timeline and looking at the length of time between Hooke first using the term cell. Robert Hooke saw cells from Quercus, from the oak tree that we make cork. So Quercus is the name, that's where cork comes from. And they're very large cells. So looking at a cork, he saw what looked like cells. He named them cells, like individual units. And that term stuck, but it took another 200 years before we recognized that as a cell as a basic unit of living function. Hi, Tammy. So you came in. We're going through our learning objectives, and if you have any questions, just stop me and unmute yourself and jump in. Right. While I'm sharing my screen, I can't see the chat or blocks, then you see it. So just jump in, unmute yourself and ask if you have any questions. So cell theory, knowing Schleiden and Schwann, these were two researchers that Schleiden recognized that plants are composed of cells. Schwann recognized that animals are composed of cells. Kind of got together, coordinated, said, hey, look, living things are all composed of cells. Rimac and Virchow, this is really interesting. It gives some historical perspective and shows this sort of thing still goes on in science. It always does. Science is very competitive. Scientists do not always play nice together because you make your reputation by proving something was, or by disproving someone else's work, demonstrating they had errors or they were wrong, or taking credit. In the case of Rimac and Virchow, these two gentlemen actually worked together in the same lab at one time. And they had both published, but first, Rimac had published a paper saying all cells derive from previously existing cells. He went through to actually show cell division. He was the first one to see, recognize, and describe cell division, that this is how we get more cells from a single cell. 3 years later, Virchow published similar paper with similar results. And he used the term, all cells arise from cells. And he was the one who got all the credit. He published more widely. By today's standards, we would consider this plagiarism because he really took word for word quite a few of Rimac's own observations. The standards and the legal component for plagiarism as theft of ideas in someone else's work had not really been firmly established at this point in the mid-1800s. But really, Virchow gets lots of credit for stuff that he plagiarized directly from Rimac. And so now textbooks are going back, and we have the insert in the textbook that talks about this. I don't think it's the ethics in biology, but it's one of those inserts that talks about this. It's quite interesting. So yeah, you should make sure you do read that. Where is it? Oh, it's science and plagiarism. It is the I on ethics portion. And some of the things that contributed, sadly, that continue to be an issue that science deals with and overcoming it is part of the reason Remak didn't get credit for that is he was Jewish. And there was a lot of anti-Semitism in society at that time. And so politically, racism raised its ugly head, and Remak kind of got screwed in the deal. So this is really a point that science and the scientific community continues to deal with and struggle with and try to atone for past slights. We see a lot of this with female researchers, and we learn a little bit about this when we talk about DNA, of men taking credit for other people's work. So that's just an interesting insert in there in the Ion Ethics you might want to take a look at. The endosymbiotic theory. This is another one. It's explained really how eukaryotic complex cells, the eukaryotes, that are complex and have compartmentalization, were able to expand and grow larger. How did that happen evolutionarily? And the endosymbiont theory talks about how these larger cells that have separate organelles for energy, the mitochondria to get energy, or in plants for organisms, where they can make their own energy through sunlight. Those organelles actually are about the same size as bacteria. They have similar membrane components to bacteria. They have a double membrane. So if I was going to engulf something with my membrane through endocytosis, and it had a membrane, I'd have a double membrane. Their DNA is molecularly more similar in the chloroplasts and mitochondria have DNA, and it's more similar to bacterial DNA. The enzymes that are used to replicate it that they contain are more similar to bacterial DNA. They have their own bacterial ribosomes. So when this was first proposed back in the 1800s, it's kind of poo-pooed, and then it kept coming around and more evidence. So another example, in 1960, a researcher, Lynn Margulies, really started amassing evidence and published that, this theory really, we've got all kind of evidence for it. And she was sort of patted on the head and dismissed. Because, what's this little girl researcher know? Turns out, once we got the right molecular techniques to look at the DNA, to look at the ribosomes, to look at the mitochondria and the chloroplasts at a molecular level, turns out they really are similar to bacteria, but they formed a partnership, this symbiotic relationship that evolved into a single organism and those organelles. So that theory now has loads and loads of molecular evidence, physical evidence, the DNA, the process of the DNA being passed on, how they replicate through binary fission, all that support that. And we go through a little bit more history with Semmelweis. This always fascinates me, that washing hands was an earth-shattering revelation discovery at one time. That, oh, look, if we wash our hands before we go into the labor room, the babies and the mothers are healthier. How about that? So Semmelweis was hand washing. Jon Snow, not that Jon Snow from Game of Thrones, but Jon Snow, father of epidemiology, he tracked the source of a cholera outbreak. Again, Pasteur also looked at pasteurization, keeping and killing microbes as a way to preserve food, and those same microbes are actually able to contribute to disease. Lister made great progress. Listerine is named in his honor, of establishing aseptic technique, using phenol as a cleaning agent, a disinfectant agent in surgery to prevent the spread of germs. And then again, his Koch postulates, he's the first one to establish a method to connect and prove causation. This is the microorganism that causes this disease. So they all contributed to this to the development of germ theory that it's not bad air that makes us sick. It's not punishment from God that's making us sick. It's microbes that cause disease. All right, so that's background on cells and the cell theory and cells causing disease. And 3.3 goes into prokaryotic cells. 3.4 is all eukaryotic cells. We don't go into that, but everybody should know that. That's us. Those are our cells. So we should know their structures and functions. What's new is going into more detail on our bacterial cells. And these structures, these are going to be very important. Remember, prokaryote, no nucleus, simple cells. We don't have compartments. Have a single cell. Everybody did great on the quiz describing cell morphologies, our shapes. So coccus are round, spheres, cocci, bacilli are rod-shaped. So we can be round, we can be rod-shaped, more elongated, or we can have spiral. So the vibrio are just a curve, like a C-shaped curve, or spirillum. They're going to be twisty, like a corkscrew, and our spirochetes. So the spirillum and the spirochetes, the difference are our spirochetes are more flexible and our spirillum are more rigid. And we don't actually look at any of those in class, but we do talk about them in lecture, our spirals. And then, so morphology is the shape of the individual cell. And then the arrangement is how they can group together. They might not, they might just be single cells, but we might have them always divide and stay together in pairs. You would put diplo in front of that, diplobacilli, diplococci, diplococcus. They may do that twice and give a group of 4 cells, tetrads. They may form chains. We use the prefix strepto, streptococcus, a strip of spherical cells, streptobacillus, a strip of rod-shaped cells. Staphylo is a cluster, so if we get big clumps, we really just use staphylo with cocci. One thing to be aware of is those terms streptococcus and staphylococcus. How we write them matters, because those are genus names. So if the S is capitalized and the word is italicized, we're talking about a specific genus of bacteria, Streptococcus or Staphylococcus. But those words can also be in regular font, not italicized, not capitalized, and then they are just adjectives. They describe a group of cells, because we do have other genuses and species that might be spherical, cocci, that occur in clusters. So they're Staphylococcus, but of the genus Micrococcus, as an example. So watch how the word is spelled or used, because it could indicate a specific organism by genus name, or it could just describe the cellular morphology and arrangement. Let's see. We have a missing bullet here. This should be a separate bullet. How prokaryotic cells maintain morphology, including during changes in osmotic pressure. Our cell wall, we have a nice, firm, rigid cell wall, and that's what allows these cells to maintain their shape. All right? And we look more at the cell wall in just a minute in a lower bullet. Osmotic pressure, our activity for week 2 when we talk about cell growth looks at osmotic pressure and osmosis. But it starts, we talk about it in this chapter. We talk about it again in growth. We talk about it again in control of growth, because osmosis is the movement of water across the cell membrane. And our cell can either take water in, if it's in a hypotonic solution. It can lose water if it's in a hypertonic solution, or it can just maintain its shape in isotonic. In all of these, that membrane, that cell wall is rigid, so the cell keeps its shape. What changes is inside that plasma membrane, the water we're talking about moving in and out goes into the cytoplasm, And so the cell membrane can pull away from the cell wall, or it can press up against the cell wall as more water enters the cell. But the cell wall is going to maintain the same shape. So what's going on inside the cell could be swelling or shrinking, but that wall is pretty firm, so only so much water can go in. So we'll look at that more in activity 2, but you should have a general idea. Water always moves toward the hypertonic solution. So the saltier solution, or the solution with more sugar in it. We'll go into that in more detail. Structure function, should know the nucleoid region is where we find our DNA. Plasmids are extra chromosomal DNA. Ribosomes, cytoprotein synthesis. Our inclusions, even though we don't have membrane-bound organelles and compartmentalization in bacteria, we can actually make little temporary holding compartments. And these can either have a protein exterior, they can have a phospholipid monolayer, just one layer of phospholipids, unlike the membrane that's a bilayer. But they hold or store nutrients a lot of times, sometimes gases. And that storage has to do, back with osmosis, water moves toward high concentrations. So if I just have a high concentration, let's say, of glucose inside my cell, because, hey, look, I'm in a good energy source, while that increase in glucose inside raises the concentration of glucose, that makes the inside of the cell hypertonic and would lead to water coming in. So to just keep an isotonic environment on either side, I can just package those nutrients or package whatever material that is into this separate housing, just to store it, to hold on to it. So inclusions are just kind of little temporary storage bodies, storage units. And then endospores, we went through the sporulation process. We talked about endospores. These are for protection. It's basically a cell going dormant, protecting itself. Builds a little protein coat around the essential pieces, the DNA, maybe a few enzymes and ribosomes. And I isolate those off. I let the rest of the cell disintegrate. I am no longer metabolically active. I'm not getting energy. I'm not using energy. I'm not replicating. I'm not doing anything. I am dormant until conditions around me improve. So until there's food or water or whatever the hazard is around me is gone, I am just going to stay in my little bubble. And then when they come out of the endospore, when environmental conditions are more appropriate, and they come out of their endospore, we call that germination. They return to an active cell state. Plasma membrane is our phospholipid bilayer. Membrane transport, passive ways that molecules get in and out of a cell. So diffusion, molecules just diffuse from high concentration to low. No energy required, right across the membrane. Osmosis is specifically water moving across that membrane. You can have facilitated transport, which is also passive. Molecules only move from high concentration to low, but through a membrane protein. And then I can have active transport, where I'm going to move molecules against a concentration gradient. I'm going to require energy. I'm going to need energy to do this. So I might have membrane proteins that act as pumps or carriers that require ATP to function. I might use endocytosis, where I bend wrap my plasma membrane around what I want to bring in and then bring it into the cell. If I want to get rid of things, I can push them out of the cell through exocytosis. Group translocation in bacteria is where I'm going to bring in a specific molecule into the cell. Let's see, what page is group? Let's see, all those are on. The transport mechanism. Oh, yeah, you don't have page numbers. So this is in section 3.3. Go down away, but that plasma membrane, knowing the components of the plasma membrane, one of the things that students get wrong more often than anything is confusing and misusing the words cell wall, cell membrane, and glycocalyx. Make sure you know the differences. All cells have a plasma membrane. That is the layer that differentiates inside the cell from outside, that phospholipid bilayer with embedded proteins. Bacteria have a cell wall of peptidoglycan. So cell wall, protective, firm structure that is not metabolically active. It serves as a structural support. Let's see, so the cell wall, we have two different types in bacteria. They're both made of peptidoglycan. Those are repeating sugar protein units that are connected by amino acids. I think about this like a railroad, so the repeating sugar protein, NAG and NAM, are like the rails, and then they have little amino acid crosslinks, little short peptide chain crosslinks holding it together. So then just put lots of those next to each other, and there's a single layer, and stack those on top of each other. We get multiple layers. Gram-positives have thick cell walls, lots of layers of peptidoglycan. Gram-negatives have just one or two layers of peptidoglycan, and then they have another membrane. So they have their plasma membrane. That is part of the cell. That is not the cell wall. It's part of the cell, the plasma membrane. The cell wall is exterior to that. In gram-positives, lots and lots of layers of peptidoglycan. In gram-negatives, just a couple layers of peptidoglycan with a lipopolysaccharide layer outside. I think lipo, that's polysaccharide, sugars. The lipo portion, it's similar to the phospholipid bilayer. It's like another membrane that's outside. One of the components of that is lipid A, the lipo part. And lipid A is a structural component of that membrane. We kill that cell if we give somebody with a gram-negative infection antibiotics. The cell dies, doesn't put energy into maintaining its organization, its structure, so it falls apart, and it releases that all those components of that plasma the outer membrane and the cell wall. It all just falls apart, and that lipid A gets free of the cell, and it's now toxic. So it is an endotoxin that can cause fever. And then we want to know the structure, the glycocalyx. This is our outer, not all cells can produce this, but some do. It's an outer jelly-like substance, a polysaccharide. And if it's very well organized, it's nice and firm and tightly bound to the cell. It's called a capsule. It's loosely bound, less organized. It's a slime layer. It's the basis for biofilms, for things like plaque on our teeth, are just layers of this all stuck together. It's a polysaccharide, so it's sugar, it's sweet, it's, it sticks, it's sticky, so it lets these organisms stick to surfaces and stick to each other. And our immune system sees this as... Did you have a question?
I'm sorry, I was just at home. I was switching to my laptop.
Oh, okay. Yep. Just unmute yourself, jump in if you have questions about any of these. We run through them. Fimbriae are just small little thread-like projections, and there's lots of them and used for attachment as well, just like the glycocalyx. It lets them get kind of tangled up together, and so they can form biofilms that way, or it lets them get tangled into the cilia. That's how a lot of bacteria that cause respiratory illnesses will have fimbriae, these thread-like structures that get interlaced with cilia in our respiratory tract to get established. Pili is a modified fimbriae. It's a single long thread-like structure that's used in conjugation. With bacteria, that conjugation is not sexual reproduction. Conjugation is just a way for two bacteria to come together, and one says, hey, look, I have this cool little gene here, this cool little piece of DNA. I'm going to make a copy and send it over to you. So that pili pulls in one bacteria to the other so they can get close to each other and then share a little piece of DNA, which you learn more about that in chapter 10. And then flagella. Bacterial flagella are structurally different from eukaryotic flagella. So eukaryotic flagella move in a whip-like motion. Bacterial flagella are anchored between that cell wall and plasma membrane, and they work in a rotor-like fashion, rotate. So more like a propeller. All right, so the bacterial structures, again, we don't go into the eukaryotic structures because we come in assuming that's one of our prerequisites, is that you've had a biology class that talked about eukaryotes, that you know our cell structures, so you should know the organelles, their structure and function. We do when we talk about disease and when we talk about our immune system, we're looking at our cells and the interactions between bacterial cells and our cells. So we do need to know, be familiar with those organelles. So do take a look and review that. Chapter 5, the eukaryotes. It's our interesting make you squirm chapter, looking at all the different infections. I will say, if this chapter really grosses you out and you find it just disgusting and horrible, you might want to consider a career that doesn't involve this as your daily life. A lot of these infections are very, very common. You're going into vet tech, lots of parasites. So I was pretty excited about the discussion, how y'all got into it. Usually get a lot of people that are like, let me go disinfect myself. So yes, watch the monsters inside me. That's lots of fun. So you know our general characteristics. Here we are looking at eukaryotic parasites. They do have a nucleus. They have organelles. They are more similar to our cells than they are to bacteria cells because we are all in the same domain of eukarya. So our general characteristics, the eukaryotic parasites are protozoans, single-celled organisms. They do have all the organelles that we have and a nucleus. The modes of reproduction, we're talking about the modes of reproduction in our eukaryotes. We can be very complex or not so complex. Many of the protozoans have life cycles that include a trophozoite. This is the adult, mature stage that feeds. So if you think trophic levels in a food chain, that has to do with where I feed. Zoo, like zoology, the animal. This is the feeding animal. And the cyst is the dormant stage. A lot of times when they're encysted, this is how they infect people. They're encysted, they're present in the water supply, in the soil, or somewhere. That's how we get exposed to them as a cyst. And then when they get into a happy place, like our gut, they emerge from that cyst to become the feeding organism. Modes of reproduction, some of these reproduce sexually, some produce asexually. Asexual would be similar to binary fission. I'm just going to make a copy of my nucleus and the DNA, and then I'm going to divide, so mitosis. So binary fission, because there's not male or female, so asexual. Budding, they make a copy of that DNA and they round it. It just kind of breaks off. Schizogony, this was one of the words, schizogony. It's a funky word. This happens with a lot of these organisms. Rather than one organism, make a copy of the nucleus and DNA, go through cytokinesis, divide into two. Then those two grow, divide. A lot of these organisms can ramp up infections really quickly because a single organism, well, that's not a big threat. Your immune system's not going to pay attention to that. But its nucleus will start to divide. Make a copy of the DNA, it'll have 2 nuclei, then those will each make a copy, it'll have 4 nuclei, and those will make a copy. Your immune system sees one organism, whatever, don't care, not a big deal. And then all of a sudden, cytokinesis happens, and if I had 8 nuclei in there, I now have 8 individuals that are going to go through that process. So they can increase their numbers really quickly through that process. But that's nuclear division without cytokinesis until I'm multinucleated and have lots of nuclei. So those are all asexual. You can have sexual reproduction, which is conjugation. Talk about conjugation in bacteria, which is just vertical gene transfer. I'm just going to move genes. It is not sexual reproduction in bacteria. In prokaryotes and eukaryotes, conjugation is. There is a male and a female, or production of male gametes and female gametes that come together, produce haploid gametes, and those two gametes fuse to form a diploid new organism. So male, female coming together. So there's true sexual reproduction with conjugation in prokaryotes and protists, in protists, sorry. But in bacteria, conjugation is just the term that we use to exchange pieces of DNA horizontally. So make sure you know which one you're looking at when you see that word conjugation. And then the reproduction, some of these organisms are able to do both sexual and asexual reproduction. So if there's another organism around, we can have sexual reproduction where each organism contributes some DNA. But if not, I'll just go ahead and do asexual reproduction. The diseases, we went through these, everybody described them. So if you just go down this list, just a quick checklist to know which organisms. I had to do the breakdown of what domain, what kingdom, their whole taxonomic classification. The really important part to remember, domain, kingdom, and then this. So a lot of those, the words that they use is describe what their mode of motility is. So remember that Entamoeba, Niglaria, they move through pseudopods. They're amoeboid. So the amoeboid, that streaming of the cytoplasm and then pulling the rest of the cell structures along behind. So they're amoeboid. The non-motile or sporozoan parasites are Plasmodium, which causes malaria. Cryptosporidium, this is one of the most common causes of gastrointestinal disorders that's associated with public swimming pools. Toxoplasma, this is the parasite, the protist that's passed through cat feces, through mammal feces. This is why pregnant women are told not to clean the litter box, because that can pass through feces. This is very, very common. It's probably everybody who's ever owned a cat or a dog has been exposed to this, has had this, is immune to it, will experience no symptoms. As adults, as children, this is not an issue, but it can cause a lot of serious problems. It can cause blindness. It can cause a lot of health issues for developing fetuses, including inducing a spontaneous abortion or resulting in fetal death. So even though this is really this non-issue for any living organism, for fetuses, it's a problem. And this is why for fetal development. So this is why pregnant women are told, don't go change the litter box, stay away from it, because it won't bother them, and they probably have antibodies. They're probably immune to it, to its impacts, but developing fetuses are not. We only have one ciliate that causes disease, so Balantidium. This is our one organism with cilia. And then the flagellates, those that move with flagella, we've got Giardia, Trichomonas, Trypanosoma. So if we remember, let's see, Trichomonas vaginalis. You remember the full name for that one? Oh, there we go. That causes vaginitis. Our trypanosomes, these cause sleeping sickness. Giardia, this is really, really common for people who are very outdoors oriented, go camping a lot or hiking. This is also called Hiker's disease. Causes, again, gastrointestinal disorder, diarrhea. Comes from contaminated water, usually freshwater sources, contaminated with feces. So mountain streams, if there are farms nearby, any kind of streams. If you're out hiking in Western North Carolina, beautiful, wonderful mountains with nothing and nothing anywhere, it must be fresh, clean water. Well, there are pig farms in those hills and water runs downstream, and that's how we get infected. But it's very common. And so if you are going into nursing, you'll see a lot of these because many of them are very common, usually not. As we get to when we get to our immune system, you'll all be very happy to know. We're probably exposed to all sorts of these, but they don't usually cause problems. I mean, we have a very high load of the parasites. So with all of these, we call them infestations, and how serious it is, how bad the infection and symptoms are, has to do with the load. How many of them do we have? We're usually exposed to these in small numbers all over the place. So those are our protists. In 5.2, we look at our parasitic worms, our helminths, which these are covered, these are macro. I don't need a microscope to see tinea, to see a tapeworm that can grow into up to 3 meters. The reason these are covered in microbiology is because the way we get infected is usually with the eggs, and so microscopically, and then they grow. So the morphology, the names, the common names of the groups tell you the morphology. We have nematodes, which are roundworms, trematodes, which are flukes, kind of leaf-shaped, and our cestodes are the tapeworms. Those are the morphologies that we're looking at. The terms monoisius and dioecious, well, these trip people up because they're not what they sound like. Monoisius, one body, dioecious, 2 bodies. And so what we tend to think of is mono is 1. Monoisious have 1 body with both sex organs. So monoisious is 1 body, has male and female sexual reproduction organs. We could use the term hermaphrodite, but that refers to a specific part of how they can reproduce with themselves and others. And then dioecious, I have 2 bodies. I have males in 1 body and females in others, so they're separate. Or reproductive organs are separate and separate separate individuals. And then in our discussion, we went through some of the different more common infections. And the reason these are here, these are brought up, is because these are the ones really in healthcare you're going to see very commonly. Ascaris is a really common infection that we see especially in poorer communities and in migrant communities. Went through enterobius, so our pinworms are really disgusting. Kids get them all over the place. Trichinella, this is why you always get told, oh, cook your pork, make sure you check the thermometer, and the meat thermometer is above 165 degrees. This is the one we get through eating undercooked meat. And then a lot of people chose Dracunculus because of that name. How can you not? Dracula. So yeah, read over the different descriptions of these that you did in the discussion. They don't all come up, but kind of best way to do this is really, draw out a table, group them so that kind of have some idea. If you see them, you remember. If you know a couple of each one, if you just know 1 or 2, sort of helps you narrow it down. So if you don't see one you recognize, pick the other answer. And our flukes, flatworms, are flukes. The biggest thing with flukes, they have really, really complex life cycles. Many of them have like 2 or 3 intermediate hosts that they have to go through in a specific order to reach reproductive maturity. And then our cestodes are our tapeworms. Everybody's pretty familiar with tapeworms. We just now have a specific name for it, a little more detail. So I would suggest, yeah, just fill out that table, the blank table that I included. Motility, what group they fall in. These are all our eukaryotes. So are they helminths? Are they protists? Let's see, and then our fungus. And our fungus, mycology, we see myco, we see this prefix a lot. Usually we're going to associate it with fungus, but our mycobacterium aren't fungus, they're bacteria. So make sure you get the whole word. Don't just look for that prefix again, just like with pro. So mycology is the study of fungus, and a mycosis is a disease or disorder caused by a fungal infection. Then we have fungus that fall into the category of yeast, which are single-celled, or molds that are multicellular. The components, so the terms hyphae, if we have multicellular, these are the long threads. So those are the hyphae. Easiest way to picture these are just think of bread mold, because we've all seen bread mold. The hyphae are the fuzzy threads. These can be septate, meaning there are lots of individual cells separated with a cell wall, a whole string of cells. Or we can have non-septate, where there's not a cell wall. There's maybe a little part of a cell wall between them. That just makes it easier to move nutrients back and forth along the whole length of the hyphae. Pseudohyphae are those that have partial cell wall going through. And if we get a whole mass of those hyphae together, it's mycelium. And the mycelium, that's when we see a whole fruiting body or a whole body of fungus that we're looking at. That's the mycelium. So it's all these hyphae packed tight together. And then our many fungus are dimorphic. And that di, two, and morph, morphology, shape. So dimorphic, a lot of fungus can either be yeast, single-celled, or they can become molds, multicellular and spread out. And a lot of times it's environmental triggers like temperature or pH that will determine which shape they're going to be. And this is important because there are a lot of infections that we deal with in healthcare, like Aspergillus, which can be a single cell, a yeast, and we can inhale that or inhale the spore. And when that gets into our lungs or in our body, says, oh, I'm pretty happy here. I am now going to grow. And so it will become this multicellular form big globules in the lungs. So some can occur either way. And usually how we're going to be infected is through a single-celled, either a yeast, a single-celled fungus, or a spore. Getting to spores, again, we're going to see that we saw spore with endospore, and when we talk about bacteria, a lot of times casually, we'll use that term, spore, but we mean an endospore when we're talking about bacteria. In bacteria, an endospore is a protective structure. When we're talking about fungus, spores are reproductive structures. Right? So these are reproductive. They're going to spread through the air, and then they are going to germinate into complete fungal cells. Let's see, the cell wall of fungus. So our bacteria have peptidoglycan. Our plants have cellulose. Our fungus, their cell wall is made of chitin. And chitin is a carbohydrate, like the same way cellulose is in plants. And cell membranes, these are eukaryotes, so all of these have phospholipid bilayer for their cell membrane. Animal cells have cholesterol as part of not cell wall, cell membranes. Animal membranes have cholesterol, and what that cholesterol does is it maintains the consistency. So our cell membranes are kind of fluid. They have the consistency of olive oil, and cholesterol does that in our cells. Fungal cells have ergosterol. They have a different type of sterol that has that function of maintaining the consistency of the cell membrane. And the reason we point out these differences is because if we're going to target these with a medication meant to eradicate them, we don't want to harm our own cells. So we have to find where our cells, because these are all eukaryotes, so our cells are very similar. So we have to look for differences so that we can come up with ways to eliminate them that target these cells without hurting us. Again, discussion group broke these down into some of the different molds and the infections they cause. Candida albicans, this is the #1 cause of yeast infections, of fungal infections in humans, is Candida albicans. It causes vaginal fungal infections, oral thrush, all sorts. And this happens when we get rid of our good bacteria. This is part of our normal fauna. It's just kept at low numbers until we eliminate it by taking antibiotics. We eliminate our bacteria. Pathogens, what fungus do. All right, so that really, coming up with tables, looking over those discussions, that's going to be the most helpful portion for chapter 5. And look, we're already at 12:36. We can go over a little bit. So biochemistry of the genome, deoxyribonucleotides, DNA. The structure of our nucleotides are building block monomers, a 5-carbon sugar, deoxyribose, attached to a phosphate and a nitrogenous base. So those bases in DNA, adenine, guanine, thymine, cytosine. And the way they pair, that anti-parallel structure, means we stack those one on top of the other, and then we make another stack, but in the opposite direction. So anti-parallel. RNA, ribonucleotides are RNA. Ribose is the 5-carbon sugar with a phosphate attached. And the nucleotides, the nitrogenous bases associated with RNA are adenine, guanine, cytosine. We have uracil instead of thymine. So thymine is never going to be found on RNA. And actually what I think I'm going to do is jump to the exercise you did to activity 1. And we'll finish that because that's going to go through most everything on here. And the portions it should be pretty familiar to you. Anyway, just replication. Replication, make a copy. So DNA has to make a copy. Transcription, translation, the different types of mutations. So let's jump to activity 1. Right, we'll go through that. I think that will be more helpful for going through these 2 chapters. Okay. So I have to open that first. Let me grab that file. Thought I had that open already. All right. Okay, share. All right, there's our activity. All right, so this is when we talk about anti-parallel. This is what we mean. We stack one set of nucleotides going from the 3 to the 5 direction, the other from the 5 to the 3. What that means is if I look at my sugar, Right, so I have a pentose, I have a 5-carbon sugar. And the way I think about that 5-carbon sugar is that it's kind of pentagon shaped. And I count the carbons 1, 2, 3, 4, and then the 5th carbon is actually off the top, 5. So by counting those, 1, 2, 3, oh, there's my 3rd carbon. So as I'm not going to keep folding these, but as I stack these to make my nucleotide, the 3s are always on the bottom, the 5 is always above it. So this one runs 3 to 5. The anti-parallel one, 5 to 3, the sugar would be upside down. So if I counted those sugars again, 1, 2, 3, 4, 5 is on the bottom now. So 5 to 3. So I'm running one set this way, the other set, that's not exactly a very good fold. So they're next to each other like this. And then connecting in between are my bases. So that's what the anti-parallel is. The sugars run in a different direction. And then my base pairing, thymine in DNA, thymine always pairs with adenine. Cytosine always pairs with guanine. If you think of this like a puzzle, that's the only piece that'll fit. So here's my anti-parallel with complementary base pairing. This is my original DNA. Number one, everybody tries to make this so much harder than it is, replication. In DNA replication, what would happen is each of these would separate, and then my DNA polymerase would come in and take this strand here and copy it, paste it here, and add new bases, the matching bases. So what replication would do is, oh, I separated this out and separate those two strands because that's what my helicase does, is it unwinds and separates the two strands. And then DNA polymerase comes in and says, okay, there's the 3, so I'm going to start at the 5' end, and I'm going to match. T gets an A, T gets an A, C gets a G, and I'm just going to do complementary base pairing and replicate this, the original, make a copy. DNA polymerase is going to come in and say, oh, here we go, I can only work this way, 3 to 5, so I'm going to do this in the opposite direction. But that's what I'm going to do. When I replicate this, it means I copy it. I make 2 copies. And everybody will usually answer this right, semi-conservative. It's because this was the original DNA up here, and this was the original DNA up here. So my 2 new copies each contain, I have conserved half of the original molecule in my new molecule, in each of my new molecules. So that's just replication. I make a copy. People want to make that harder and use different base pairing and try to build RNA, but replication is used because when I need to go through binary fission for a bacteria, I need to increase my number of cells. Well, to have all the directions, I need all my DNA, so I need to make a complete copy of my DNA. So now I started with this much DNA. I make a copy so I can divide it. This cell gets some, this cell gets some. Everybody has all the DNA. So that process happens every time we need to get new cells.
I have a question about that.
Yes.
Wow, echo. So we're making a copy of it, which kind of means that we're leaving the original there, but we're really not. We're actually taking one of the originals and then matching it. So we're not left with the original DNA strand at all.
Right. Because in binary fission, I'm going to have 1 bacteria, and then when I'm done, I'm going to have 2. Right. So I'm never actually going to have like mom and dad, and then here's baby, or mom is going to have a new offspring. Mom's going to make a copy of everything inside her and split into 2.
Okay. So we're not copying that strand. We're just kind of separating it and moving them, and then matching the bases in the opposite direction to each one.
Right.
Because we're not left with.
Yeah, so we are making a copy of the original molecule. We're making two copies of the original molecule. So that we start with one, we end with two, but the way we make the copy is we split this into two and we use it as a template. So a template is just like, it's a pattern.
That's what I thought, but I thought we were going to split it and then copy this line and then make the other half to go with it, and that didn't make any sense to me. So it doesn't matter if we're splitting the 3 to 5 and the 5 to 3. We're copying both of them regardless, so I don't have to choose one over the other.
Right.
Okay.
We're going to split them in this one, so the 3 to 5 becomes a template to build the 5 to 3. And the 5 to 3 from the original becomes a template to build this. And the reason we have to build the opposite, that we can't just make a direct copy, is because of the enzymes. So enzymes can work one way and only one way. And our enzyme, DNA polymerase, can only read this going 3 to 5. And it can only, if it sees a T, it can only put an A there. So it couldn't, make another T, make another T, make an exact copy. It has to put the complementary base pair. And then this other one, because it can only read 3 to 5, it has to build this one in the other direction. So here it jumps on, it latches here and says, okay, add this, add this, add this, add this, add this. On this one, it says, here's what I'm using as my template. I have to start here and go this way. And the
That's where the lagging strand comes in.
Yes, because we'll look at our chromosomes because we're used to them in their threads. When the helicase opens up the DNA, it doesn't open the whole thing and leave it there. It opens a little at a time. So once to 3 to 5, it can go zoom as it opens. This other one, it's like, oh, I can only do this because that's the only part I have access to. Oh, now I can do this because I have access there. Right, so that's why we get that lagging strand.
Okay, got that.
And then here, DNA to protein. So our central dogma we take DNA, we transcribe it to RNA, we translate that to a protein. And we do that through complementary base pairing. Only now, when I have an A in my DNA, I can't get a T when I make my RNA because T's don't come in RNA. I can only get my U's. So here, we have the template strand, which is used as a template to build RNA with complementary base pairing. The other strand, so here this tells me this is my template strand. So here's my template strand. This is what I'm going to build my RNA from. This is called the coding strand, which sometimes trips people up because wait, if that's the coding strand, isn't that the one I'm going to use? And I explain in those hints why it's, or in the follow-up, why it's called that. Because when I build my RNA from this template with complementary base pairing, the RNA has complementary base pairs, so it ends up with these base pairs. So the RNA looks identical to the coding strand, but that's not what I built it from because that's not how RNA polymerase works. It can only match complements. So, but if I want to do a quick check, did I make my RNA correctly? If this is my template, I can look at this strand and say, oh, it should be identical, except I'm going to put a U here. So it's a good quick check, but that's why that's called the coding strand. So here this question asks, what do you do first? You transcribe first. I have to get RNA. So my T becomes an A, my C is a G, my G becomes a C, my A now has U, so C, U, C, and then A gets a U, C gets a G, T still gets an A. And then I would translate by going to the codon table and looking up those codons. Here, we say that the code is degenerate. We have multiple options for getting the same amino acid. There are 64 possible combinations. So if we jump down and look at this table, we see, oh, there's a lot of repetition. And if you look closely, where you see that repetition is, as long as I have the right first two bases, I'm going to get the right amino acid. So the fidelity, or how true I have to be to the codon, degenerates from the first to the second to the third base. By the time I get to that third base, whatever, throw anything in there, I'm still going to be okay. That gives us some protection from mutations. So I can have a mutation and still be okay, those silent mutations. It doesn't change anything. So that gives us protection against mutations in the DNA and also changes in the RNA, because RNA polymerase, when I transcribe this, DNA polymerase had proofreading ability, so it could go back and correct its mistakes. RNA polymerase doesn't. So if it makes a mistake, we've got a little bit of protection against having the wrong amino acid in place. This one's not directly addressed in the book. I just like to see what people think as they go through there. The primary reason, DNA is a big, giant, huge, double-stranded molecule. Our DNA contains all the instructions for every single thing our cells do all the time, or that they can do throughout our lifetime. We don't need all that all the time. We need one protein right now. I need this enzyme to do this job right now. So RNA is a smaller molecule. Instead of all of the chromosomes, all of the DNA, it's going to make a copy of this much of it. It's going to transcribe this piece. And it's single-stranded, so it's free to move out of the nucleus out into the cytoplasm where I can build my protein. We don't want our DNA jumping out into the cytoplasm all the time. We want to protect it because it's important stuff. So it stays in the nucleus, doesn't go anywhere else. And that's to protect it. So we use this intermediate to get out to the ribosome. Let's see. So our codons are found on our messenger RNA. And I go through and I recite this every time. My messenger RNA has the message in code that tells me how to build my protein. So I know I have to go to the codon table with my messenger RNA codons. These are my triplets of nitrogenous bases that tell me which amino acid I get. So that's the mRNA. The anticodon, anti-not. This is not the codon. This is not what's in the table. Don't use this. What this is, right. So the tRNA, it's RNA, so it's made the same way. So it has those base pairs. And what the anticodon is, it's the complement to the codon. But the reason is because it works like a lock and key. Right, those complements, my C can only connect with G, nothing else. So here's my strand of mRNA and my 3 codons. Well, here's all these tRNAs, they don't know what to do. So any one of them could come in there, but only one matches. Only one fits. So the anticodon is the correct fit so that I get the right amino acid in the right place. And then our RNA, this is kind of confusing, but the ribosome is actually made. The ribosome, the site of protein synthesis, is made out of RNA wrapped around proteins. It's like, oh, boom, which came first? The ribosome? Oh, the ribosome's made of ribosomal RNA. And proteins that are made at the ribosomes. But yeah, that's what it is. It's a structural component of a ribosome. Just my ribosome has RNA and it has proteins. And that's what makes those two subunits. So that's just a structural component of it. Okay. So transcription, translation, the difference. Everybody did that well. That part of the ribosome. Proteins. Everybody wants to say, oh, strong bones and teeth, drink your milk, yay, hair and nails, and structural. Bacteria don't have any of those things. The really, really critical role of proteins are their enzymes. So enzymes speed up chemical reactions, and metabolism, the sum total of all the chemical reactions in a cell. Every single chemical reaction that takes place in every cell, all the cells in our body, every bacteria, all those chemical reactions are sped up by enzymes. So without those enzymes, metabolism wouldn't happen at a fast enough rate to sustain life. So we need those enzymes. So we need those proteins for anything that we do.
Okay.
All right, so our puzzle, this is just a fun puzzle in this chapter, but this becomes really important that you understand this when we talk about viruses. In eukaryotes, in bacteria, living organisms, we have double-stranded DNA. One strand is the template strand, and that strand always makes RNA. We always have tRNA that's also built from somewhere from DNA, same way, but this carries my amino acids, brings them in. This is just here to trip you up and try to get you to translate the anticodon. This is the one that we want to use on the table to get our amino acids. So we always work in this direction, DNA to RNA to amino acids. When we get to viruses, viruses think this is a fun game. We have viruses that come in. In viruses, we don't have M and tRNA. What we call it is positive and negative RNA. So we have viruses that might have this strand of RNA, and from this, they can directly make a protein. Or they might come in and with this strand of RNA, like HIV, a retrovirus, and make the DNA. So if they have this, then they can build this strand of DNA. And if they have that, they can build this strand of DNA. So they can work either direction. They can come in with this strand, the negative messenger RNA, that doesn't tell me how to build a protein, but because of complementary base pairing, if I have that, I know how to make that. I know the base pairs, and then I can make my protein. So bacteria can come in with one strand of DNA and still get, they can come in with this strand of DNA and still get to here, right? They just have to make this strand first and then this strand, and then they make their protein. Or they can come in with this strand and go directly to protein, or to mRNA, to a protein. So viruses play this game. So not understanding the base pairing. So here, no matter what set of bases you have, you can work up and down. So even if I know this DNA, well, I know it's complement here, so I can tell you what the mRNA is going to be. I can tell you what the tRNA is going to look like. Even though it's the anticodon and I don't need it, I can tell you what it is because of base pairing. So any of these I can just work. All I had to do from here was say, well, tRNA has the anticodon, the opposite base pairing, so I can move up here and do this. And then from here, I know what DNA code, what DNA triplets would do that. So that was just going up and down between the lines until you filled it all in. But you really don't even need that one. And I think the sickle cell, I think this is really interesting because we think of these diseases, they must be horrible, awful, all kind of changes, and it's one single amino acid that changed right there. Instead of glutamate, we got valine. And how do we do that? Well, here's glutamate, GAA or GAG. All I need to change that is get a U by mistake. One little base pair mistake. And we get that whole trickle down explosion of related problems because of it.
Okay.
One of the questions in the discussion was about why bacteria have so many different methods of horizontal gene transfer. So vertical gene transfer is parent to offspring, generation to generation. Horizontal, I can't go and say like, gee, here, you want some of my genes? Have some. These are good. I have good genes. Let me make a copy and give them to you. But bacteria can do that. They can say, oh, here's a good gene. I'm going to make a copy and share it. So we have all those different methods that they can share those that you discussed, the 4 different ways. And, the reason I went to is because bacterial replication is cloning. So every bacteria of the same species would be an identical clone other than the background rate of mutation, which is really low, unless they had some method to get variation. Whereas eukaryotes, we have sexual reproduction, mom, dad, we get a mix of genes every time there's an offspring. Every new offspring has new genes. So we have all kind of variation in populations. So bacteria get that variation through gene transfer. And that variation is important because if every organism is exactly identical genetically, then they're all susceptible to the same threats. So they might all be fine as clones in one environment, but if the environment changes to something that's lethal, they're all going to die. Whereas if there's variation, they're not. So if a few of the organisms have a resistance to antibiotics, and most of them don't, but if you have that, well, now I'm going to take an antibiotic because I want to kill that bacteria. Some are going to survive as long as there's variation. But if they were all clones, they would all die. So if one has a mutation that gives it the benefit of like, hey, look, that antibiotic doesn't bother me. Let me make a copy of that part of my gene and make a little plasmid here and I'm going to share it. So that's why they have all that. They have to they have to find a way to mix up their genetics to get variation, since their their replication doesn't do that. And then the operon, one of my students, I thought, came up with the most brilliant analogy. Like, what's an operon? Why is this so complicated? Because you don't need all your genes all the time, just like you don't need, so he needs to an app on a phone. You would just use up all your resources, all your minutes, all your data plan, and you would use up all your battery life if you ran every app on your phone all the time. So and that would do the same thing, bacteria or any cell would just use up all its energy if it just expressed all its genes all the time. So this is just an on-off switch. So operon is out here. I don't need this right now, I'm not going to use it. And so what it does is when the environment is right, that I need this now, oh, lactose is present. Here's my lactose, my inducer, it's present. I need this now. So let me combine with my regulator, it'll fall off. This is my RNA polymerase. Now I can go and make, these are my genes, RNA polymerase makes messenger RNA so that I can make the protein that digests lactose. So I can use that for an energy source. Lactose isn't really abundant. Glucose is more abundant. So I want the genes that break down glucose all the time, because that's going to be my most abundant sugar source. But if all of a sudden I find I have lactose instead, I'm going to turn those off and I'm going to come make my enzymes that break down lactose. But once the lactose is all gone, right, it would break down this lactose too. That lactose is all gone. And now this comes back in here and shuts off my operon. It really is like a roadblock here. Now my RNA polymerase can't go down and transcribe my RNA, so I'm not going to make that. So it's really on-off switch so I can be energy efficient. I'm only going to make the proteins, the enzymes that I need right now. And when I don't need them, I'm going to turn this off. So we have constitutive genes. Those are the ones that I always need to do this. I always need to make these molecules. They're always on. Then I have repressible genes. These are ones that I usually need. Like glucose is usually present. I usually need to have my glucose enzymes so that I can get energy. But if glucose isn't present, I'm not going to make those. So I can repress those. And then inducible. Well, lactose is not always around, so I really don't need to have the enzymes and keep making those enzymes if that food source isn't available. So I induce them, I turn them on only if lactose is present. All right, any other questions?
There was something, and I can't find it in my cards now, about, I thought I had written it down, but I can't find it. Something has single-stranded DNA. And I don't remember what that was or how do you have single-stranded DNA?
So when we get to viruses in chapter 6, we'll talk more about single-stranded DNA because viruses have that. Let me see if who else has a single-stranded DNA? Yeah, it should be viruses have single-stranded. Some viruses. Viruses either have RNA or DNA, and some have double-stranded and some have single-stranded.
Okay, maybe that's what I read somewhere.
So yeah, it might have started to refer to that because we usually have in bacteria, it's double-stranded, but it's in a single ring. So it's one ring, but it's still two strands.
Okay, gotcha.
Where we have 46 chromosomes or thread-like DNA. So going over that, I know this is a lot, especially if it's been a while since you've learned any of this, and then you have to,
Yeah, I don't remember any of this from when I took this class. Like, well, I mean, not all of it, but I took this class, 12 years ago, and it was not, I don't remember this stuff being in there. I mean, maybe it was, and I did better back when I was younger, but trying to think. I think my only other question was a lab question that's not even on this test. But maybe you would know the answer to it while I have you. I see that we're not doing like staining techniques and stuff. That must be just a lab thing. But I was trying to remember the capsule staining, and what I read was some sort of a red dye, but this says nigrosin. I thought it was in one of your quizzes, but maybe it was in one of the lab quizzes.
it was probably in the lab quiz. And there's actually two different ways to do a capsule stain, where one stains the capsule one color and the cell another. But the capsule with nigrosin, it actually repels it, so you end up with it's a negative stain. So usually you stain the organism and you see the organism against a white background. The negative stain, what happens is that capsule repels the stain, so you see a black background. It looks like looking at a starry night because the organisms are white. The organisms are all clear. So that's what the nigrosin does with the negative stain.
Okay, so for our class, we're not using the red one as the primary. We're using the nigrosin then, because I think what I found on Google and what I got wrong on one of the quizzes, I think it was nigrosin on the quiz, but what I had found under Google was some red one or something. So I just wanted to make sure I had the right. It was
the other one we use is methyl blue. Yeah, with copper sulfate with the blue.
Okay, so that's not how we're okay.
Yeah,
we're
so
nigrosin and methyl blue with water. Okay.
I've got a spreadsheet that actually you probably have that. It's a spreadsheet with the different stains, and it says, we do this alternative, but here's the other stain. And I don't know if your instructor put it in your classroom, but I'll put it in the lab classroom. But I'll put it into our lecture classroom. I'll post that. Okay. We're coming up in our next unit. We're going to talk about microbial growth, which is really just going to go into detail on a lot of the different things that you're doing in lab, because you're growing bacteria in lab. So there will be a lot of coordination on that. What you're doing in lab, what you're reading about and doing in lab with that chapter.
So on this test that we're taking in this class, we're going to have a portion where we have to explain two things. I guess we get to choose from four things, but there's two things that we have to explain the whole process of, right? something.
So there are 4 questions. You can pick any 3 to answer. You can answer the 4th as a bonus. And they're on different, there's some compare and contrast. One's a compare and contrast. Think about the cell wall. I ask about the cell wall. I ask about, you think I could remember what I'm going to ask you, couldn't you? Cell wall. Oh, if you look at the discussion questions, I pull a lot from the discussion questions. So there's probably something about prokaryotes. How would you identify prokaryote versus eukaryote, maybe? Something about the differences there. Maybe label the lac. Yeah, label the lac operon. Here's the picture. Here's your picture of the lac operon. Label the parts. And I can't remember what the 4th one is, but something like that.
Gotcha. Okay. I think that's it. I think I'm pretty good on it. We hope. So I'll probably take it later on tonight.
I think it becomes available at midnight tonight, I think. So it's Friday till Sunday. So you have all weekend to take it.
Awesome. Okay. Well, I think I'm good. Thank you so much.
All right. You're welcome. Thank you. Email if you have any other questions.
I will.
And I'll post the recording of this. I will, post once it processes. Takes a while to process, but it'll be up probably in a couple hours. It'll be in the classroom.
Thank you.
All right. Thank you.